Hi, Mrs. Melchatter. We're going to go through some cell membrane notes, chapter 5 in our book. So here we go. This is a picture, a cartoon image of what a cell membrane would look like. And you can see that it's a pretty active place that has a whole bunch of other things in it, through it, and on it. So we're going to talk about all of these different components that you would see, like the glycolipids and the proteins and the cholesterol. So let's start with the structure of a membrane. We have talked about this already, but just as a reminder, a membrane of most cells and also inside of the cells in the endomembrane system will be phospholipids arranged in a bilayer. So 2 layers with the hydrophilic heads facing out towards the watery environment or towards the outside of the cell, and also facing towards the inside of the cell. And then those hydrophobic tails turn towards each other and away from the water. Inside of the membrane are these globular proteins that are in between different parts of the phospholipids. Some of them will be kind of on the top and hanging a little bit down. Some of them may even go all the way through. And we call this the fluid mosaic model because it's like this mosaic. It's all these different little pieces, just like a mosaic painting or a picture would be a bunch of little pictures or pieces put together to make a bigger image. That's what we have with the phospholipids and the membrane. We have a bunch of little stuff that makes this big picture. So these proteins and lipids and cholesterol, they're going to float in or on that lipid bilayer or in between. And it's very fluid and it moves. It's not just a stationary, hard structure. Just kind of cool to think about because when we look at our bodies, we can't see those cell membranes moving. So we've also seen this structure. We went over it in our POGL, but this is the phospholipid structure. So up here, we've got an amine group. Here's our phosphate with the PO4. That one's going to have the negative charge, positive up here. That's going to make that polar. Then we've got our hydrophobic tails at the bottom here with those long chain of carbons and hydrogens. Okay. And then to the right is just the cartoon drawing of what we envision that as. So we have the membrane, and we also have this thing called the ECM, or extracellular matrix. You will see it referred to as ECM all the time and rarely as extracellular matrix, but that's what it means. And it's this area that's between 2 cells. And it's always going to be pictured as this really big area. We have to remember that our cells are really, really close together and even tied together at certain points. And all of this stuff is going to be between them. And that's the extracellular matrix. So there's 4 main components to the plasma or cellular membranes. And I don't want to just say cellular membranes because then we get the idea that it's just the ones surrounding the cell, but it's we also have membranes on the mitochondria that are going to have all of these different structures and pieces too. So there is the phospholipid bilayer. Those are the little hydrophilic heads and the hydrophobic tails that we've talked about before. They're flexible, movable, and this is going to provide a barrier. So this is going to, with that polarity, is going to control or determine what can enter through the cell or not, through the membrane or not. Then we have transmembrane proteins and integral membrane proteins that are, so transmembrane proteins would go all the way through the membrane, so from the outside of the cell to the inside. And then you have the integral proteins that will be inside the membrane itself. You have an interior protein network, which will include peripheral and intracellular membrane proteins. So proteins that are on the outside or the periphery, and then proteins that are hanging out just on the inside of the cell membrane. So on the bottom part into the cytoplasm. And then we also have a whole bunch of cell surface markers, which are going to include things like glycoproteins, glycogen protein, and a glycolipid. And those are going to basically be able to mark your cells as liver cell or this person's cell. This is your cell, and this is part of your body. When we study the cell membrane, they can look at the cell membrane not with a light microscope, unfortunately. So if we look at cells under the microscope, our microscopes, we can just see a thin outline, especially if the cells are stained, of where that membrane would be. But our scopes aren't strong enough to actually get and look and see those phospholipids in those areas. But you can with a transmission electron microscope, or TEM, and also a scanning electron microscope, or SEM. One method that they can use is to embed whatever membrane that they want to look at in an epoxy, and which is, I think, almost like a plastic kind of ish thing. And then they're going to shave off 1 micrometer, so really, really, really tiny thin layers, and then you can see. So here we have the cell membrane of one cell, and then the plasma membrane of another, and here's that ECM, that intracellular matrix. Okay. And this is a pretty small sample. 25 nanometers is just that area right there, so it's not that big. Another way that they can study cell membranes is called the freeze fracture microscopy, and that's going to help to see the inside of the membrane and also to see where the proteins are embedded and which parts of the plasma membrane those proteins lie in. So they basically take a cell and they freeze it in a medium, in a substance, and then that is cracked with a knife blade. And what will happen is that membrane will fracture right in between those two layers of the lipid bilayer. And when it does, when it fractures and separates, it does so that the proteins that are embedded in one side of it will remain in that side. So we can see, like, oh, this one would be a protein that might be peripheral, or this one would be a protein that would be on the interior of the cell, or this one would be a transmembrane protein because it seems to go all the way through and there's a space in the other side, like here. And they also have to coat the membrane with platinum, which makes it like a replica. Okay, a little bit more here on the phospholipids. So as a reminder, that phospholipid consists of a glycerol, 2 fatty acids, which are those tails there, and they always show one as unsaturated and the other is saturated, but they don't necessarily have to be that way. There is those fatty acids are nonpolar and hydrophobic, and then the phosphate group that's attached to the glycerol that part is polar and hydrophilic. And if we were to just dump these into a liquid aqueous environment, they will spontaneously form a bilayer because those hydrocarbon tails will want to be flipped inside and protected, and the polar heads will be on the outside. We need to understand that the bilayer is very fluid and moving. Even though we seem solid and we are solid, if we were to look at the membrane, those phospholipids will change places with each other, and they move very fluidly. There is only weak hydrogen bonding that's holding the two layers together, and also weak interactions between the phospholipids that are holding those together. So you will have these individual phospholipids, and any proteins that aren't anchored or bonded to part of the phospholipid that will be able to move around and move through the membrane. Some things that influence fluidity, or the ability of phospholipids to be able to move. Well, so saturated fatty acids, those are going to be the ones that have no double bonded carbon. They're going to make the membrane less fluid. There's no kinks in the chains of the fatty acids, and so they're going to hold it tighter and closer together, whereas the unsaturated fatty acids are going to have double bonds and be a little bit more kinked or kinky, and that's going to hold them further apart. Also, warm temperatures, our membranes will be more fluid than cold temperatures. And that's where we'll stop for today, and we'll pick up with the rest on the next video. Thank you.